So today we're going to start by finishing chapter 23. We have two sections left. We have 23.8, which is focusing on the liver, and then we have 23.9 looking at regulation of food intake. Then after we finish 23, we'll move on to chapter 25 for reproduction. And for that, we're just going to look at meiosis because it's common to both male and female reproductive systems. And then next week, we'll actually separate out, we'll have the male reproductive system, and then we'll have the female reproductive system. Okay. So on to the liver. The liver is an amazing organ. It carries out over 500 metabolic functions. And as I've mentioned, we have machines that can mimic a heart. They can mimic the lungs. They can mimic the kidneys with dialysis, but we actually do not have a machine that can take over the role of a liver. Okay. We're going to be looking mainly at how it regulates plasma cholesterol levels, but it also metabolizes alcohol, drugs, hormones, bilirubin, ammonia, and turning into urea, as we know, and as well as storing vitamins and minerals. This is a summary of most of what it does, or some of what it does. For the carbohydrate metabolism, the things we're looking at, are going to be, it stores glucose as glycogen. And skeletal muscle can do that also, but the liver, that's one of its primary roles. And then it will perform glycogenolysis to release that blood glucose into the blood when blood glucose levels fall. The liver is the primary site of gluconeogenesis, so converting amino acids and glycerol to new glucose. And as we'll investigate more, converting glucose to fats for storage and sending those as VLDLs to adipose tissue for storage. For fat metabolism, it's one of the primary sites of beta oxidation. Cardiac muscle, skeletal muscle are also going, and adipose tissue are actually all going to be sites of beta oxidation, but it is a site of beta oxidation. So the breakdown of fatty acids to acetyl-CoA can convert acetyl-CoA to ketone bodies, which we didn't really go into ketone bodies, as well as synthesizing lipoproteins for transport of fatty acids, fats, and cholesterol, and synthesizing cholesterol from acetyl-CoA. For protein metabolism, remember we have that amino group, that NH2, that we have to take off of amino acids if we are going to use them and oxidize them for fuel. So one of the key roles of the liver is to synthesize urea in order to remove ammonia from the body. So these are all three of these, the amino group, ammonia, and urea are all nitrogen-containing compounds. So we need to be able to excrete those from the body. We can't allow them to build up. Deaminate or transaminate amino acids, that's what, going back to getting rid of this amino group so that they can be oxidized or converted through gluconeogenesis for glucose. But again, we need to get rid of ammonia, and we will use it to synthesize urea so that can be excreted. The liver also stores vitamin A, stores vitamin D and B12, and it stores iron. It metabolizes alcohol, drugs, and other toxins by tagging and activating them and marking them for excretion. It processes bilirubin and excretes that as well, or tags it for excretion. And that's about it for the summary. So we're going to be looking a little bit more at cholesterol. There are three types of lipids. Okay, so we had triglycerides, and these were used for energy. Okay, so we broke it down into glycerol. Glycerol gives about 15 ATP, and then 3 fatty acids. Each fatty acid will produce over 100 ATP. So triglycerides are the only class of lipids that are really used for energy metabolism. Then we have phospholipids. And this is a glycerol plus 2 fatty acids and then a phosphate group. It makes it ideal to be used in that phospholipid bilayer of the plasma membrane. So this is the plasma membrane lipoprotein. It is rarely used for energy, like if it is, needs to be degraded or reused, those fatty acids could be, but usually we're synthesizing phospholipid. We're not really using it for energy. So that's phospholipids are for plasma membranes. And then the third category of lipids is going to be cholesterol. And cholesterol is going to serve 2 big functions. So one is that it's going to add rigidity to the plasma membrane. Okay, so that's where we get this major component of plasma membranes. Phospholipid makes it fluid. Cholesterol makes it rigid because it's branched and it gets in there and it inserts itself. And so our cells need a balance of both in order to keep some of the proteins in those membranes kind of where they should be. The other thing about cholesterol is that it is going to be used in steroid hormone synthesis. It is actually the starting point of all steroid hormones. So all steroid hormones start at cholesterol, and then we can build testosterone, we can build estrogen, we can build progesterone from there. But they all start with a common skeleton. It is also used in bile salts, and we'll talk about how unsaturated fatty acids cause excretion of cholesterol and bile salts. So that's one way to lower cholesterol, but I'm getting a little ahead of myself. Okay, so cholesterol is not used as an energy source. Okay, it is used in bile salts, steroid hormones. We're not going to go into vitamin D, and plasma membrane rigidity. 15% of blood cholesterol is ingested, and 85% is made by the liver. So cholesterol is almost like largely derived from acetyl-CoA in the liver. So that is the main source of cholesterol. So the main source of cholesterol is acetyl-CoA in the liver. So the liver metabolizes acetyl-CoA and forms cholesterol. That accounts for 85%. Okay, so now we're talking about lipoproteins. So one of the roles of the liver is to produce lipoproteins, which will transport cholesterol, phospholipids, and triglycerides through the blood. The higher the percentage of lipids, the lower the density. There are 4 types of transport lipoproteins. This one, we're going to go with chylomicrons first. These are not produced in the liver. They're produced somewhere else. Does anyone remember where chylomicrons are produced? In what type of a cell? No takers? Okay. So in the small intestine, triglycerides are going to be broken down into glycerol, monoglycerides, and free fatty acids. So it gets broken down, and then it gets transported into the cells of the small intestine. In the small intestine, these get reformed. Okay, so we've got monoglycerides, fatty acids, and glycerol, but they get reformed into triglyceride, packaged as chylomicrons and exocytosed into the lacteal, that green structure. Okay, so chylomicrons, these are going to come from the small intestine. These are transported from the small intestine. They have the lowest density and consist almost entirely of triglycerides. The liver is going to produce these. And on the next slide, I get into their functions, but they are named by their density, and fat is not density. So for VLDLs, these are the very low density. They have the most fat. Low density lipoproteins are kind of in the middle. They have the highest cholesterol. HDLs, the highest density of the three lipoproteins, is going to have the highest protein content. So you should be able to match these with their characteristics. So, and you also need to know their functions, okay, or their purpose. So very low density lipoproteins transport, because they're really high in triglyceride, so they're about 50% triglyceride. And yeah, we will talk about, we're going to talk about cholesterol levels. So VLDLs are going to transport triglycerides from the liver to peripheral tissues, mainly adipose, for storage. So this is going to repurpose or repackage the triglyceride from the chylomicron into VLDLs, send it back out. If it's not going to use it for beta oxidation, send it out to the adipose tissue for storage. LDLs are the lousy type of cholesterol, of lipoproteins. They are the ones that are high in cholesterol. So LDLs transport cholesterol to peripheral tissues for plasma membranes, storage, or hormone synthesis. When tissues need cholesterol for the membranes, they'll synthesize LDL receptors, they bind the LDL, and they bring the cholesterol into the cell. That's how it's supposed to work. But when we get high LDLs, then it starts incorporating into the blood vessel membranes. So there's so much cholesterol floating around, it starts sticking or embedding in other membranes because it is a lipid. So remember, lipids are lipid soluble. If you get too many of them, they're not controlled. They are going to find a place to hang out. Okay. So on to HDLs. HDLs are the healthy. I'm going to actually change this to green. These are the healthy ones. Because the liver actually produces the envelope, the lipoprotein envelope, the proteins, and they're flat, and it sends them out, and then they fill up with cholesterol from other tissues. So this takes cholesterol from peripheral tissues or blood vessels to the liver to be broken down and secreted in bile. Okay, so HDLs are healthy, LDLs are lousy. So you want high HDLs and low LDLs. Okay, I'm going to come back to this one in just a second. but we're on kind of on a roll with cholesterol levels. So you should be able to tell me if something is a good cholesterol, total cholesterol level, a bad total cholesterol level, a good HDL, a bad HDL, same with LDLs. We know when you go and you get, if you're at my age, they take your blood about once a year and they tell you don't eat anything before you come in because we want to take your LDL level and see what that looks like. I mean, not your LDL, take your cholesterol level and see what that looks like. So you go in, you get your blood taken, and the first thing they're going to look at is total cholesterol. So total cholesterol levels should be 200 mg per deciliter or less. Over 200 is linked to atherosclerosis and heart disease. However, more importantly, they will look at the HDL and LDL levels. HDL is protective because it pulls cholesterol out of membranes, so out of, well, not really membranes, but out of cells and out of cell walls, and it takes cholesterol out of the blood. So somewhere between over 60 would be great. Less than 40 is not good. Then LDL, so you want this as high as possible. And LDL you want as low as possible. So 100 or less is good, 130 or above is not as good. So if you had 100 here and you had 60 here, your total would be 160, and that would be great because you have high HDL, low LDL, and a low overall total cholesterol. This is just a nice little picture. It does show you, one of the things I like that it shows is that every single, so these are lipoproteins. These are the three categories of lipoproteins, but chylomicrons also. All of these have triglyceride, phospholipid, cholesterol, and protein. So they're going to, on this outer membrane, it is made of proteins and phospholipid. And then in the middle, you get triglyceride and cholesterol. So triglyceride and cholesterol hang out in the middle because they are subclasses of lipids. You will get a little bit of cholesterol in the membrane, just happens. And then, but primarily the outer membrane is going to be made out of protein and phospholipids. So protein and phospholipid on the outside, cholesterol and triglyceride on the inside. All of these have those same four components, just in different percentages. VLDLs have the most triglyceride because these are mainly going to adipose tissue for fat storage. So chylomicrons come from the small intestine to the liver, bringing triglycerides from what we eat. The liver is going to store some fat. It will do some beta oxidation to oxidize fat, but then what it cannot use, it packages as VLDLs, sending the triglyceride to adipose tissue for storage. It also manufactures LDLs and HDLs for cholesterol management. So chylomicrons, VLDLs, really focused on triglycerides. LDLs, HDLs, these are going to be your cholesterol devices. LDLs bring cholesterol from the liver to tissues. They are lousy or bad. HDLs take cholesterol from tissues back to the liver for excretion in bile. So these are your healthy. Your book also does a really nice job. It's not too long and it's not too complicated. And the liver, like these questions kind of make themselves. So it's really easy to make questions from this section for exams. Factors regulating blood cholesterol levels. So the liver is going to produce cholesterol at a basal level regardless of dietary intake. Restricting dietary cholesterol does not markedly reduce blood cholesterol levels. However, the choice between saturated fatty acids and unsaturated fatty acids will have an effect on overall cholesterol level. Unsaturated fatty acids, so these are going to be plant oils, are going to enhance excretion of cholesterol into bile salts. Okay, so that's why they're always telling you eat more unsaturated. So go with olive oil, go with canola oil rather than butter. Although personally, growing up in the South, you eat a lot of butter down there and bacon. So, but plant unsaturated fatty acids are much healthier. Saturated fatty acids stimulate liver synthesis of cholesterol and inhibit cholesterol excretion from the body. So the balance between these does have an effect on cholesterol levels. So eat more plant oils and less butter, or fatty meat falls under there as well. That is 23.8. We are moving on to 23.9, regulation of food intake. For this, the hypothalamus is going to be in control of feeding behavior. It has something called an arcuate nucleus. So this is the group of neurons that is involved in feeding behavior. And they have two populations, and actually has more than that, but three that play a role. But these, it has groups of neurons, let's say that will either, one group will produce signals. Neuropeptide Y is the big one that enhances appetite. Says, okay, time to go grab something to eat. And then there is another population of neurons that is going to secrete peptides, POMC and CART, that will suppress appetite. You don't need to actually know the names of the peptides. Okay. You do need to know that the hypothalamus is the major player in regulating food intake, and that it has both populations of neurons. It has a population that enhances appetite, so makes you hungry, and it also has a population that suppresses appetite. So the hypothalamus houses both of those populations of cells, so it is the controller. There are also going to be signals from the vagus nerve and the enteric nervous system, and stretch receptors in like the stomach. Okay, so there are neural signals, nutrient stores, rising blood glucose or amino acid levels inhibit appetite, as well as rising fatty acid concentrations. For hormones, we have hormones that stimulate appetite. These are going to be glucagon, epinephrine, and ghrelin from the stomach, as well as hormones that inhibit appetite, such as insulin, cholecystokinin from the small intestine, PYY, which we never talked about, and leptin from adipose tissue. From this slide, there are two things to take home. One is that the hypothalamus is the major player in feeding behavior because it has the neurons that regulate appetite enhancement, as well as the neurons that regulate appetite suppression. However, there are other inputs from neural signals such as the vagus nerve, nutrient stores such as rising blood levels of glucose, amino acids, and fatty acids, as well as a hormonal input. Okay, so you should be able to pick these out as well as factors that influence feeding behavior. Short term are the 3 factors that I just talked about. So short term regulation of food intake are going to be neural signals from the digestive tract. The main one is going to be the vagus nerve. There are also stretch receptors in the stomach that will send signals as well. But for short-term regulation, the main neural signal is going to come from the vagus nerve. In addition, if you increase blood glucose, amino acids, fatty acid levels, these are also going to suppress appetite. So you should be able to know if something is suppressing appetite or not, I guess. For hormones, we're just going to lump these together as a category, okay? But they are insulin, cholecystokinin, glucagon, epinephrine, ghrelin. So hormones can either stimulate appetite or inhibit appetite. Rising blood glucose, amino acids, fatty acid levels, these are all going to inhibit appetite. And then the vagus nerve is also going to inhibit appetite. Long-term, so the previous 3 were short-term regulators, so on an hour-to-hour basis. So leptin is an interesting hormone. This actually was discovered when I was in college. At least that's when I first heard about it. And they thought it was going to be this revolutionary breakthrough and make us all skinny. They thought they were going to be able to prescribe leptin and we would all lose weight. And then it didn't work out that way. And people were very disappointed because it looked like the magic bullet for weight loss. Leptin is secreted by adipose tissue. And so this is why they thought it would be the magic bullet, because as you get more fat cells, you get more leptin. And then this is going to decrease appetite. So researchers thought all we have to do is give people leptin, it's going to be an appetite suppressant, and everyone's going to get skinny. But it turns out that it didn't work that way, that it is an indicator of total energy stores in fat tissue, but it actually only comes into play when it's lowering, not elevating. So it's going to protect against weight loss in times of nutritional deprivation. So not the magic bullet, but leptin is involved in long-term regulation of food intake because it is secreted by adipose tissue and is an indicator of total energy stores in fat tissue. We have a lot of fat, we get a lot of leptin, it tells our brain we have sufficient energy stores. This is a diagram that puts it pretty much all together, and that we have short-term controls of food intake regulation. These are going to be the vagus nerve. So increase vagal activity, we end up getting appetite suppression. Okay, so that's the end result here. So these first 3 here, okay, are all following this pathway. So this kind of shows you, if you want more information, how it all does flow in through which neurons. You're like, I know, just knowing the hypothalamus is good, right? Okay. So short-term controls, vagal afferents, high blood glucose, amino acids, fatty acids make their way along this pathway and decrease appetite, as well as the hormones insulin, cholecystokinin. Again, I'm not going to separate out which hormones are appetite suppressants, which ones are inhibitors. For me, hormones get lumped in together, and that we have some hormones that suppress appetite, and we have some hormones that enhance appetite. Okay. But your book does a better job than I do, and it is going to parse them out as it should, be an informative source. That we have ghrelin from the stomach, glucagon from the alpha cells of the pancreas, epinephrine from the medulla, adrenal medulla, go to a different set of neurons, and they enhance appetite. Long-term control, the one that we are concerned with today, is leptin. Leptin is produced by adipose, and it gives us an indicator of our total energy storage capability. We're not going to go into insulin right now because of insulin dependent diabetes and NIDDM. That's all left. Hopefully you learned about that in endocrine. Okay. So our long-term control is going to be leptin produced by adipose tissue to give you an idea of long-term energy storage. Additional factors in the regulation of food intake, temperature. Usually we go into thermoregulation, but we did cut that out this semester because I feel like we just don't have time to go into it. Stress depends on the individual. Some people will be, when they get stressed, they eat. Some they get stressed, they don't eat. So it can go either way. Sleep deprivation, sleep will make you hungry. Lack of sleep, will make you hungry. And then this is a really interesting new area of research, but it's turning out that the composition of gut bacteria is probably a key player in maintaining body composition. Okay. So that finishes off chapter 23.